So my colleagues and I looked at the effect of direct eye contact in PTSD related to interpersonal trauma, an fMRI study of activation of an innate alarm system. So our study evolved from the crucial role mutual eye-to-eye contact plays in social interactions and nonverbal communication. We know that direct eye contact is a very complex and rich source of information processing that allows an individual to identify and differentiate between facial expressions, so happy, sad, angry. This process is assumed to occur automatically and with little cognitive effort and has been referred to as the theory of mind or mentalizing. When we make direct eye contact, this has been found to increase emotional experiences and create greater emotional expression. Now, flip this idea to individuals with PTSD related to childhood abuse and the implications mutual eye contact has for them. Individuals who have experienced interpersonal trauma throughout childhood tend to abstain from making direct eye contact, as this interaction is known to provoke abuses and is often thought of as threatening. So does this in turn mean that they are inadvertently skipping the mentalizing process? Previous studies have hypothesized that individuals with PTSD may not be able to distinguish between positive and negative emotions due to abnormal brain activation when interpreting facial cues. Direct eye contact is instead thought to automatically initiate an alternate process in those with PTSD, the subcortical pathway or our innate alarm system. So in short form, it is hypothesized that when a healthy individual makes direct eye contact, this activates the fusiform gyrus, superior temporal sulcus, medial prefrontal and orbitofrontal cortices. So these brain areas are associated with the process of mentalizing or reflective cognitive processing, basically a healthy individual reading social and facial cues. In those with PTSD related to prolonged childhood trauma, however, mutual gaze is thought to instead activate the superior colliculus, periaqueductal gray, pulvinar and amygdala, or the innate alarm system. So this is what we were wanting to investigate, these two separate neural processes in response to the same stimuli. So we were interested in mapping out the neural pathways in brain areas associated with mutual eye contact between a group of 16 healthy female controls and 60 females with PTSD related to childhood trauma. This was done using an existing virtual reality tool from Schrammel and colleagues back in 2009 and adapted to fMRI. Our hypotheses were healthy controls would respond to direct eye contact with increased activation in the brain regions associated with mentalizing, so the DMPFC, TPJ and TP. In contrast, we expected individuals with PTSD to respond with activation in the brain areas associated with the subcortical pathway, so the LC, SCPAG and the amygdala. Participants were placed in a whole body MRI scanner along with a head coil and instructed to watch a video sequence attentively. We played 48 video sequences in randomized order, showing a 3D animated virtual man walk into the center of the screen and engage in one of the 4 different combinations of body posture and gaze, along with 3 possible emotions that I have displayed up on the screen. So we used only male characters as they were rated slightly higher on how natural they appeared, as well as their dominance and sociability over the possible female characters. While the video sequences were taking place, we acquired bold fMRI images from the participants for each combination of stimuli. And the only significant effect between the experimental groups was the bold fMRI patterns of the congruent and incongruent contrast. So there was no difference in response regarding emotional gaze direction between the 2 experimental groups that we found. So in the box on the left, we have the region of interest showing increased activation during direct eye contact with congruent body posture of the virtual character. So as you can see, there was a significant variation in cortical area responses between the 2 experimental groups. So the 3 bold images on the left are the control subjects with their DMPFC, right TP and left TPJ showing increased activation, whereas the PTSD group shown in the 2 images on the right, one on top of the other, responded with an increased activation of the left SC, PAG and bilateral LC. So in the box on the right are the transverse slices during brain activation. Again, we can see that the activation of the prefrontal cortex and other brain areas associated with mentalizing in red for the controls whereas the PTSD group shown in yellow displayed an increased activation predominantly within the subcortical areas. So our hypotheses for both the control group and PTSD group were supported by the findings. The healthy subjects responded to direct eye contact with activation in the brain areas associated with mentalizing. When we compare this to averted gaze, eye contact clearly reflects evaluative processes and provokes higher level social cognition. We saw this in the whole brain analysis where bold responses for the control group was almost exclusively in the brain regions that we had hypothesized. These findings provide evidence to the ongoing social interaction of healthy people as the brain evaluates the mutual eye contact and associated cues. In comparison, we saw increased activation of the subcortical pathway during the direct gaze in PTSD groups, irrespective of the emotion. We found that eye contact did not activate the brain areas associated with mentalizing in this group, perhaps suggesting that evaluation of social interactions by PTSD subjects doesn't occur. It was evident through the whole brain analysis that the activation of the SC and PAG was accompanied by heightened responses of the LC. So these brain areas have been linked previously to basic neural responses such as pain, fear and anger, as opposed to complex thoughts and clear judgment and perceptions. The LC in particular is thought to play an important part in the sensory evoked responses and has been implicated in the control of vigilance in animals. These results suggest that the subcortical pathway may be related to a more general facial processing that instead detects potential signals of threat. So past studies have told us that stimulation of the areas associated with the subcortical pathway in animals, so the dorsal PAG and deep layers of the SC, evoke anxiety-related responses. So defensive behaviours like running, exaggerated startle, cowering and escape. All behaviours that can be commonly observed in individuals with PTSD. So rather than a healthy individual making eye contact, holding that gaze and assessing facial cues or mentalizing, an individual with PTSD will automatically activate the alarm system when eye contact is made. This hyper-responsiveness of the subcortical route may be linked back to frequent and sustained activation of this neural pathway during prolonged childhood abuse, leading to malfunctioning of the SCPAG and development of automated defensive behaviours. Again, because we saw that subcortical route was activated across all three facial expressions, it could be that mutual eye contact signals a threat regardless of the expression on someone else's face. So our study went one step further than previous papers, presenting virtual characters in video sequences rather than still images. I would classify this as a strength and limitation of the study in the broader context of our hypotheses. Obviously, this creates a more realistic interaction between the observer and character. However, a previous study by Sagan and colleagues found that making eye contact with a robot or biological person led to notable differences in brain activity when compared with a human. It would be beneficial for future research to include a live person in the video sequence to create a more natural interaction and response. Another factor worth noting was our presentation of body posture and gaze. The body posture and head direction were always equally aligned, with gaze averted in a number of sequences. However, a more realistic course of events would be to include a congruent head direction and gaze with an incongruent body posture. So basically the character looks as though they're appearing or looking over their shoulder towards the observer. This slight adjustment would appear more realistic because when we are looking at something, our automatic response is to shift our gaze and head position together, not just our gaze. I just wanted to make one more quick note that while we did exclude a number of comorbidities, anxiety wasn't one of them. So therefore, the occurrence of anxiety may have contributed to the overall observed effects. Anxiety is known to be associated with hyperactive orienting behaviours, avoidance behaviours, and a failure to rapidly disengage from threatening stimuli. So attempting to control for this in the future would be valuable. The amygdala is theorised to play a pivotal role in perceiving threat and emotional processing, and as such, it has been linked to the subcortical route and its automatic response to emotional stimuli. Surprisingly, we didn't observe any increased activation of the amygdala like we had hypothesised, so this suggests that the LC is mediated by a structure other than the amygdala within the innate alarm system. I thought that would be interesting to follow with research in the future, or that our current methodology and technology available to us is not sensitive enough to pick up any increased activation. working to capture the role of the amygdala in the subcortical pathway within the context of PTSD subjects would be an interesting avenue to follow, as there are mountains of literature and research on the link between the brain area and emotional threat responses. So following this line of inquiry in the future may help us to assess how the neural responses in traumatised individuals impacts on their ability to interact with others, particularly within the psychotherapy and possibly in the parenting role. Thanks very much.